Hello everyone, welcome to my first video on the paper 1 series for AS level biology. I'm going to start with paper 11 from October-November 2025 series. So straight on to the first question. It's saying in a photomicrograph of magnification 5,000 times, a chloroplast measures 25 millimeters in diameter. What is the actual diameter of the chloroplast? We know that we're going to say magnification is equal to image length over actual. Now if you want actual will be equal to image over magnification, like that. But we know we can't use millimeters, we have to convert them to micrometers. And for every 1 millimeter that we have, it's 1,000 micrometers. Right? So this will be 25 times 1,000 micrometers, which is 25,000. So what we're going to have here is the image, which is 25,000 micrometers, over the magnification, which is 5,000. And if we do the math, we'll see 5 micrometers. One of the reasons that we had to convert this 25 millimeters to micrometers is our answers. We can see that all our answers are in micrometers. So we definitely had to convert that to micrometers. Then on number 2, it says, what is the function of the Golgi bodies? And for part A, is saying formation of vesicles for endocytosis. We need to remember that endocytosis is like cell eating. So if we have a cell like this and the substance coming towards the cell, the first thing that we're going to see is a bulge to accommodate the substance. Then lastly, we're having a vesicle formed inside the cell. And no Golgi body is involved in that. Maybe if they had said exocytosis, where we have a vesicle being formed inside the cell that moves towards the cell surface membrane and fuses with it to let stuff out, but here they said endocytosis, so we are not going to take that. Then for B, it says modification of proteins for secretion. See, secretion. But before we take that to be our answer, let's move on to C, synthesis of ATP. No, because we know that ATP is synthesized by aerobic and anaerobic respiration in the cytosol, that's anaerobic respiration, which is the cytoplasm, and in the mitochondria for aerobic respiration. That's where we get our ATP from. Then synthesis of polypeptides. We know that the synthesis of polypeptides is the work of ribosomes, so this is a no, and we're left with B to be our answer. There's secretion here to guide us, and it's also good to remember that Golgi bodies do help with the modification of proteins, but I was thinking that it might not be in our syllabus to cover, that's why I do ion secretion. So answer is B. Then for number 3, it says a cell in the human body is specialized for the synthesis and secretion of lipids. Which row shows the organelles required by the cell for synthesis and secretion of lipids? And looking at part A, we have the mitochondria ticked, and the smooth endoplasmic reticulum ticked, and vesicles ticked. Remember, we're looking at synthesis and secretion, and so for secretion we definitely need vesicles and the smooth endoplasmic reticulum. That's for secretion. And for synthesis, we are also going to need the mitochondria for energy in form of ATP. You know, wherever there's synthesis, we're going to need energy, and we know we get that energy from aerobic respiration, most of it, which happens in the mitochondria. And we need the smooth endoplasmic reticulum and vesicles for secretion. So that makes A the best answer for number 3. There's really no need for us to go through the other 3 answers. So then we move on. Now, on the 4th question, we are given a photomicrograph which shows part of a cell. And the question is, which row is correct? We are having the first column saying the cell will contain 70 S ribosomes. The second one saying this could be part of a plant cell. Third one, the organelles shown could be used to make antibodies. Let's take a closer look at this photomicrograph. We can see that this is a mitochondria, and these are rough endoplasmic reticulum. We can see by the structure there with ribosomes all over the reticulum, like that. So now, for us to say the cell will contain 70 S ribosomes, it would be true since we know that we have 70 S ribosomes in the mitochondria, which is kind of similar to prokaryotes, which have 70 S ribosomes. We know for eukaryotes we have 80 S ribosomes, but for mitochondria that take this a similar form is to prokaryotes, and they have 70 S ribosomes. So it's safe for us to say the cell will contain 70 S ribosomes. So we discard C and D. Then says this could be part of a plant cell. From what I see, I don't see any structure that can tell us if this is a plant or animal cell. But what I can tell you is we find the mitochondria in both plant and animal cells. So then we move to the last column. It says the organelles could be used to make antibodies. Yes, because we have RERs, rough endoplasmic reticulum, which are lined with ribosomes, and we know ribosomes are for protein synthesis and antibodies are protein, and we also have the mitochondria for energy. So I'll go with A, which is all three ticked. And that's for number 4. For number 5, it says which statement about viruses is correct? And A is saying they are cells. Then we have to stop right there. Viruses are not cells. So B, which is also saying they are cells, we're going to discard it. Then we can see the C and D are saying they are particles. They are particles. We have the same words up until here, made of. This one is saying phospholipid and this one is saying protein. And we know that they're not, the capsids are not made of phospholipids, but rather are made of protein. So D becomes our answer. And for number 6, it's saying a solution X was tested for the presence of non-reducing sugars. It did not contain reducing sugars. Some steps that can be used to test for the presence of biological molecules are listed. Then the first step is saying add Benedictus solution to the test tube. The second one is saying add dilute hydrochloric acid to the test tube. Then 3 is saying add sodium hydrogen carbonate to the test tube. Then the fourth one is saying heat the test tube in a water bath. Then the question now says, which order of steps to identify the presence of non-reducing sugars in solution X is correct? Remember, for non-reducing sugars, we first need to break them into some sort of simple sugars, and for that we use an acid. And after breaking them apart with an acid, we then add a base to kind of like neutralize the acid before we can test for the reducing sugars that would have formed after breaking the non-reducing sugars. Then after that, we can carry on with a test similar to the Benedictus solution. So looking at our steps there, we can see that 2 is saying add dilute hydrochloric acid, which should be our first step. So we're discarding A and D. Now for the second step, after breaking them, we need to add a base, and we can see that we have sodium hydrogen carbonate to kind of like neutralize the acid. And so 3 should be our second option. So then B becomes our answer. Now on to the 7th question, saying the diagram shows the structure of part of a biological molecule, and we can see it fairly ugly. And the question is, which statements about this biological molecule are correct? The first one is saying it has monomers joined by ester bonds. Let's take a closer look. Can we call these ester bonds? Because ester bonds should show something like this, and we don't see it here. So then that means number 1 here is wrong. So then we can move straight to the answers and cross out every answer with 1 on it. That's C, B, A, and our answer automatically becomes D. And for number 8, it says which diagram has a covalent bond that joins monomers together to form a biological polymer in eukaryotes? And if we look at A, we can see that A is showing an ionic bond. And how did I know that it's an ionic bond? It's because it's a bond that is formed between 2 oppositely charged ions, with this positive and this negative, and that forms an ionic bond. But we are looking for a covalent bond. And if we look at B, B is showing us a hydrogen bond. This is a hydrogen bond that forms between oxygen and hydrogen there. And we don't really see hydrogen bonds holding monomers together to form a polymer. And that allows us to go to C. And looking at C, we can see that we have ester bonds. Not bonds rather, but a bond, an ester bond that is shown. And out of all the biological molecules that we cover in the syllabus, we do not find ester bonds holding monomers, but rather we find them in the cell surface membrane where they hold phospholipids, and those are not technically regarded monomers. And that leaves us with D, and D shows a phosphate group covalently bonded to two R groups, as you can see there, which can be sugars of adjacent nucleotides, and we know that that forms a backbone or bond that joins nucleotide monomers to form a biological polymer, which is our DNA or RNA. We can see that it's showing a phosphate group that is covalently bonded to two R groups, and this forms a backbone that joins nucleotide monomers to form a biological polymer, which can either be DNA or RNA, and that's why D is our best answer for number 8. And for number 9, this is which feature of storage polysaccharides means they do not change the water potential of cells. So for a storage polysaccharide to change the water potential of cells, they need to be soluble. And if they're soluble, then they can dissolve into the liquid part of the cell, altering the water potential of the cells, because it then decreases and water gets into the cells by osmosis. So we're looking for a feature that means they do not change. This should be like they are insoluble, which is our C. And for number 10, that's saying which molecules contain at least 4 double bonds. And let's start with a saturated triglyceride. We know that it's made up of a phosphate head and these glycerides, 3 of them forming triglyceride, and we have ester bonds here. And for ester bonds, we know they're like this, and we have double bond. So this would be 1 double bond, 2 double bond, and 3 double bonds. But here the question is saying at least 4, meaning 4 or more. So then that means this is out of the picture, and we can see that C includes that, D includes that, and A includes that. So our answer will be B, because for triglyceride we only have 3 double bonds from the ester bonds and not 4 or more. So then our answer will be B. And for number 11, it says in some people a mutation can affect the beta chains in the hemoglobin molecules. As a result, polar amino acids on the outer surfaces of the chains are replaced by non-polar amino acids. And then it goes on to say what will be the effect on hemoglobin. Now, if we have a hemoglobin molecule, for example, which is polar amino acids on the outer surface, then that means it's soluble. But if those polar amino acids are replaced by non-polar amino acids, we know that non-polar substances do not dissolve, or in other words, we can say they are less soluble. I'm getting the less soluble phrase from our part A and part A there. And I know that's going to be our answer because it's saying hemoglobin will be less soluble. B is definitely wrong, and this is a lie as well, because hydrophobic interactions have nothing to do with the polarity. And D, which is saying more hydrogen bonds will form in the molecule. Remember, we are saying the polar amino acids are now replaced with non-polar, so we can't talk about the formation of hydrogen bonds. Then for number 12, saying the graph shows the effect of substrate concentration on an enzyme catalyzed reaction with and without a competitive inhibitor. Then the question is, what is the effect of the competitive inhibitor on the maximum velocity and the Michaelis-Menten constant, Km? Then A is saying the Vmax decreases. I don't see that happening because we are reaching the Vmax with the inhibitor and without the inhibitor. We can see that both of these graphs do meet here, which is our Vmax. So for us to say the Vmax decreases would be wrong. So Vmax decreases, but we'd rather say the Vmax stays the same, because with inhibitor we get to it, and without the inhibitor we also get to the same Vmax. So it's safe to say the Vmax stays the same. Now let's look at Km. Now Km is the substrate concentration at which the enzyme performs at half its maximum velocity. So if this is our Vmax, half the maximum velocity will be somewhere here. And now let's compare the substrate concentration for the two Km values. We can see that without inhibitor we are here, and with inhibitor we are here, meaning our Km increased. So then our answer becomes C. And for number 13, it says the graph shows energy changes in a chemical reaction. And the question is, what is the activation energy when an enzyme is added? So we know an enzyme reduces the activation energy of a chemical reaction. And for the arrows to show activation energy, they start from reactants. We know our reactants will be over here and go all the way to the peak. So they should be starting here. Either we're taking this peak or we're taking this. But we can take this because it's longer than this one. We are saying an enzyme decreases or reduces the active site. So we'd rather take the smaller one, which is shown by 2. And that's B. If we take 1 plus 2, then we take the longer one. If we take 3 minus 2, then we'll be starting from this end. We always start from the reactant side, because this is the progression of reaction, meaning it started this end and it ended on this side. So we have to start from this side. That's why I assumed that our reactants will be here. And we always start from the energy level of reactants to the peak. And now for number 14, it says the diagram shows an enzyme, its substrate, and an enzyme substrate complex. Enzyme substrate and enzyme substrate complex. It says which statement explains how this substrate is able to enter, the active site of this enzyme. Because as you can see, the shape is slightly different from this. We cannot say they're complementary just by looking at them, but if we look at the enzyme substrate complex, we see that the substrate fitted perfectly, almost perfectly, into the active site. That's what the question is saying. Explain how the substrate is able to enter through this narrow active site. Then A is saying contact between the substrate and the enzyme causes a change in the enzyme shape. This is sounding a bit along the line, but let's look at the other options. The shape of the active site and the shape of the substrate are complementary. No, as you can see from this part of the diagram, they are not complementary at all. If they were, we wouldn't be getting this question to say how is the substrate able to enter. Then for part C, saying the substrate within the active site forms hydrogen bond. The question is asking us how the substrate enters, so we cannot talk about the substrate already within the active site. So that makes C wrong. And D, when the enzyme substrate complex forms. So here the question is asking us how the enzyme is able to enter, and we know that an enzyme substrate complex can only form after the substrate is inside the active site. So then D becomes wrong, and our only answer will be A. Now moving straight on to 15, saying what are the main roles of glycolipids in cell surface membranes? Then first one is to help cells to attach to each other to form tissues. 1 is correct. Glycolipids do help cells to attach to each other. Then 2, to act as antigens for cell-to-cell recognition. 2 is also correct. Now for 3, to say to increase fluidity for the cell surface membrane. We know that that's the work of cholesterol and phospholipids, so then 3 is wrong. So we have 3 here, 3 here, and 3 here. So then B is our answer. Now on number 16, it says a sample of healthy plant cells taken from the same tissue is placed in a beaker containing distilled water. The cells change size. What would explain this change in size? So we've taken plant cells, then we place them in a beaker containing water. We know that if we have a cell placed in water, we're going to have water getting into the cell via osmosis, down the water potential gradient from high water potential to low water potential, and that explains the change in size because the cell will increase in size. So A is saying water will leave the cells, which is wrong. If water leaves the cells, then the size will decrease. So then B saying water will enter the cells by osmosis. I'll go with B. Let's look at C. Solids will enter the cells by active transport. We're not talking about active transport if we're talking about water in cells changing size. And solids will leave the cells by osmosis. We know that osmosis is only for water molecules and nothing else, especially at this level. Then on number 17, saying students studying the surface area to volume ratio and diffusion made a cuboid S1 using agar stained blue with a pH indicator. Then goes on to say the dimensions of S1 are shown in the diagram. The student made a second agar cuboid S2. Each dimension of S2, the length, the width, and height was half that of S1. You can see this is S1 and this is S2. Then it goes on to say the student placed each cuboid in a test tube and covered it in acid. The time taken for each cuboid to completely change color was recorded, and all variables other than the size of the cuboids were standardized. Which row shows the surface area to volume ratio of S1 and the time taken for S1 to change color completely in acid compared to the time taken for S2 to change color completely? I think I'll start with the last part of the question, which is talking about the time taken for S1 to change color completely, because we're saying if we're looking at these two, this S1 is obviously bigger than S2. So if we're having the acid traveling towards the center of these two, it will take less time on this one than this one, and hence we are going to say S1 takes more time, or S2 takes less time. You can see that S1 takes more time is here and here. So we're discarding A and C. Now onto the surface area to volume ratio. I'm going to quickly calculate the surface area for S1. The surface area, so this side and that other side at the back, this one and this one. So it's going to be 30 times 8, which is 240. I'm going to multiply that by 2 for the other side. Then for this side, I'm going to say plus 5 times 30. Let me just put that in brackets. 5 times 30. I'm getting 630. 5 times 30, I was supposed to say times 2. I'm getting 930. And lastly, for 8 times 5, which is 40, plus the other 40 for the top, which is 80. I'm getting 860. 860 for our 30 by 8 times 2 for the other side, and 5 by 30 times 2 for the other side, and lastly 8 times 5 times 2 for the top part. Adding those, I get 860, and that's for the total surface area. And for the volume, we're just going to say length times width times height, which is our 30 times 8 times 5. 30 times 8 times 5, and we're getting 1200. So for the surface area to volume ratio, going to say surface area over the volume, and if we simplify that, 860 over 1200, we're getting 0.716 with a bar showing that it's recurring. And looking at this, we have 0.72 S1 here, which is close to this 0.72. So we go with 0.72 S1 and this, other than 1.4 S1. And for number 18, says 2 test tubes labeled X and Y with setup containing equal volumes of solution X and solution Y respectively. A large number of type P cells and a large number of type Q cells were added into test tube X and also into test tube Y. Then it says, after a few minutes, samples of the solutions were taken and the cells were observed with a microscope. It says, in solution X, all the cell type P had best and cell type Q had not best. Then in solution Y, no cells had best. Which row correctly identifies the cell type Q and solutions X and Y. So cell type Q is the one type that had cells which did not best. So if it did not best, then looking at animals and plant cells, I'll go with plant cells which have cell walls, and that way we have high chances of cells not bursting even when water is to get in by osmosis. So red blood cells will be out, goblet cells, liver cells. So then we're left with D, router cells, and the rest can sort itself out. But let me see if I can explain a bit. Solution X is distilled water. So if we put cells, two different types of cells in water, if it's a plant cell and an animal cell, if water gets into the animal cell, because it does not have a cell wall, it's going to burst. And if water is to get into the plant cell, likewise, the cell wall is going to maintain the take pressure, so the cells will not burst. And if cells are put in 5% of sodium chloride solution and said no cells had burst, it means water was moving outside of the cells due to a difference in water potential, so none of the cells had to burst. There was really no pressure to withstand. Now on number 19, it's saying a human muscle cell contains 46 chromosomes. How many centrometers will be present in a cell during how many centromeres will be present in a cell during the stages of the mitotic cycle? So for this one, I don't think there's really any reasons for us to go through all the responses. I'm just going to explain what happens during early prophase, metaphase, and anaphase in terms of the chromosomes. And if we think about what we have covered and early prophase, we know that at this stage DNA is already replicated during the S phase, and each of the 46 chromosomes consists of two sister chromatids joined together. And since we're saying they're still joined together at a single point, they count as one chromosome with one centromere. If we draw our chromosomes like this, the centromere being the central part, and if we're saying they're still joined at a single point, we can still count them as one. So it's safe to say we'll be having 46 centromeres. But now, when we look at metaphase, where we're having the chromosomes lined up at the equator of the cell, although we can say they are highly condensed and they're visible pairs of chromatids, we still know that each pair is still physically attached to a single centromere. So again, we'll be having 46 chromosomes, not 46 chromosomes, 46 centromeres, my bad. And for the last part, which is the anaphase, where we're having the centromeres splitting, then that means we're going to multiply our 46 by 2 because we are having the centromeres splitting. I think this was the tricky part of the question. And so when we have the centromere splitting, then we can go up to 92, and that's why A would be the best answer. Number 20 is saying telomeres become shorter during each cell cycle of a specialized cell. And the question is, in which phase of the cell cycle do telomeres shorten? And for this, again, before we even look at all the answers, it's have to know or to remember that the part of the cell cycle where we have telomeres shortening is the S phase, and this is because this is where we have DNA replication occurring. And the reason why the telomeres shorten is because of the enzyme DNA polymerase, which cannot replicate the very tip of the legging strand. I'm sure you remember that. So that is the reason why we're having our telomeres shortening, because DNA polymerase cannot replicate the tip ends of the legging strand, and that occurs during the S phase. And we're having the S phase on option C. Moving on to number 21, saying the photomicrographs show stages of mitosis, and says what is the correct order for the stages of mitosis. And let's look at option A. It's saying we start with 2, then we move on to 3, then from 3 we get to 5, then 1 and 4. I think A is most sensible, since it's clearly showing us how the stages go from prophase, metaphase, anaphase, and lastly telophase. And for number 22, says the gene that codes for protein R is 130 kb long, and protein R is 220 kDa in mass. The typical mass of an amino acid is 110 Da, and 1,000 base pairs is equal to a kb, and 1,000 Da is equal to a kDa. And the question is, which row is correct? So for this question, I want us to think of a gene like a movie, right? It is actual scenes. In this case, our actual scenes are going to be mRNA. These are actual scenes, and commercials, right? Which in this case are the introns, which are going to be spliced out. You know, you don't care about commercials, you only care about the actual scenes in the movie. Now, with this understanding, we're going to start by dividing this 220k by this 110Da, because this is the mass of one amino acid, and that is going to give us the number of amino acids that we have there. So we have 220k over 110, and that is going to give us 2k. So now we have 2k amino acids, and with that, I want to take you back a bit. Remember that we have some stuff that we call codons, right? Which we can say in simple terms, 3 DNA letters. 3 DNA letters which are read from our A, T, C, G. We know these letters are read in threes in order for us to form amino acids. So we're going to use that 3 for each codon that makes up each one of these 2k amino acids. Now with that 3, I'm going to say our 2k times the 3, and that is going to give us 6k. Right? So it will be 6k letters long, because we took 3 letters, the ones that make up each amino acid, then we multiplied it by the number of amino acid. So we have 6k letters, and this is for our RNA, or actual scenes for our movie. Now, looking at the commercials, the introns, we are going to say if the whole protein is 130k long, right, 130 letters long, like that, and only 6k letters are needed to make it into a final movie in our case. So we are going to say this minus the 6k to get 124,000 introns, which in this case are commercials. And I think I will have to take that again. I said, let's imagine this to be a movie, and we're going to take our mRNA to be our actual scenes and our introns to be our commercials, because we know that the introns are going to be spliced out, and we really don't care about commercials when you're watching a movie. So then I divided the 220 kDa, in which is mass of the protein, by the mass of a single amino acid, which is 110 Da, and I got that we have 2,000 amino acids in there. And like I said, we have codons, right, which are 3 DNA letters which are picked from A, T, C, and G to form an amino acid. And to calculate the total number of letters we have, those DNA letters, I said 3 times the 2k, then I got 6k, and that's for the mRNA, right? Then I moved on to the commercials. Then I said if the protein is 130,000 DNA letters long, and we subtract the 6,000 letters for the mRNA, which is our actual scenes of the movie, we are being left with 124,000 introns, which in our example would be the commercials, which we don't need. And we're going to use these numbers that we got from our working to get the answer. And we can see we have 124,000 and 6,000 on C. I hope that was clear enough. If I left out some of the parts or any of the parts unexplained, please do share in the comments below. Or if any one of you guys have a shorter method of doing this question, please feel free to share in the comments as well. Now on number 23, it says the diagram shows a section of DNA, and 2 of the bases in this section are labeled 1 and 2. 1 up here and 2 down here. Then the question says, which row correctly identifies the bases 1 and 2? And for this, looking at our answers, we have purines, pyrimidines, adenine, guanine, adenine, and guanine. And for us to be able to pick G or A, we know that A is always bonded to T, C to G, and we have 2 hydrogen bonds in between A and T and 3 hydrogen bonds between C and G. So we can see that this is 2 hydrogen bonds and this is 3. So 2 can only be G. Now, for us to determine whether we have a purine or a pyrimidine, we're going to look at the rings. And looking at these two, we can see that they both have 2 rings, and that makes them purines. And so our answer will be B. Moving on to 24, it says which role describes the role of enzymes DNA polymerase and DNA ligase. Let's run through the answers. I'll start with DNA ligase. It says it joins sections of DNA together on the lagging strand. B is also saying, no, it's saying the leading strand. So I thought it was the same all the way through to D. But B is saying on the leading strand, lagging strand, and the leading strand. And I'll take this one at the leading strand because DNA ligase joins sections of DNA together on the leading strand and not on the lagging strand. So I'll take D and B. Now, to the DNA polymerase side, it says add the nucleotides in the 3 to 5 direction, and D saying adds nucleotides in the 5 to 3 direction. And we know that DNA polymerase adds nucleotides in the 3 to 5 direction, and so 24, our answer will be B. Moving on to number 24, which says which role describes the role of enzymes DNA polymerase and DNA ligase. And if we look at our answers, A is saying DNA polymerase adds nucleotides in a 5 to 3 direction, and we know that this is correct. DNA polymerase adds nucleotides from the 5' end to the 3' end, and so A and D will be correct on this one. Now, for DNA ligase, it's good to remember that it deals with the lagging strand and not the leading strand, and so we're just going to look for the lagging strand. And there we have it, join sections of DNA together. The joining sections part is written on all our answers, but the tricky part is whether you remember that it deals with the lagging strand or the leading strand, and that's how you have to get to your answer. So the answer is A. Maybe in a separate video I'll go through the explanations in detail, but for now our answer is A. And for number 25, says the table shows some of the DNA triplet codes for some amino acids, and we have a list of amino acids, DNA triplet code, which is the 3-letter code that we talked about earlier, and amino acid DNA triplet code. It says the best sequence on the template DNA strand coding for part of a polypeptide is shown, and given CCA, TTC, ACG, GCG, TTA, and GCA. And says 2 mutations occur in this sequence during DNA replication. Which mutated DNA would result in 2 different amino acids? So let's go through our answers. For part A, we have CCA, CCA, CCA. Then on place of TTC, we have ATC. So TTC, if we look for TTC here, we have lysine for TTC. And for ATC, we have stop for ATC, and that's not an amino acid. And for ACG, let's just check ACG is the same, GCG is the same, TTG, TTA. TTA is coding for asparagine, and TTG is coding for the same amino acid, so there's no change there. And we have GCA, and we're looking for 2 different amino acids. So even if we had to consider this to be an amino acid, the stop one, it still does not meet the number 2 because we need 2 different amino acids. So A is out. Then we move on to B. With CCA, the first one is the same, TTC, ACA in place of ACG. So if we look for ACG, it's cysteine, that's ACG, and ACA, it's also cysteine, so it's the same. And GCA, we have GCG, going to compare GCG and GCA. GCG, arginine, and GCA, it's arginine as well. and now TTA is the same, GCA is the same. So now B again is out. Then we move on to C. We see CCA is the same, TTC is the same, ACG is the same. Now on the last bit, we have CCG in place of GCG. And we know that GCG is arginine, CCG is glycine. So now we have one different amino acid. But then we have TTA and GCC. For GCC, we are looking at arginine, and for GCA, it's also arginine. So we only have one different amino acid. And this tells us that we only have two different amino acids on option D, since we've gone through all the other options. And if you were to compare these, there's triplet codes with these ones, you find out that there are two different amino acids. And so D would be the answer. The diagram shows a vertical section through part of a stem of a dicot plant. 3 types of structures are labeled. And it says, what are the structures U, V, and W? And now, from our answers, we see we have the companion cells, the phloem sieve tube elements, and the xylem vessel elements. And just by looking at the diagram, we can see sieve plates. And we know that we only find sieve plates in the phloem. So phloem should be our U. And there we have it, no other U's. So I'm suspecting that B is our answer. And just to confirm, for the xylem, we have W. And you can see that clearly shows xylem with the lignified walls. And now the last one, companion cells, you can see that this is the only part that is cell-like on this diagram. This part, you can see this, which look like nuclei. So that would be representing companion cells. And for number 27, says, which features of xylem vessels help to reduce the resistance to water flow? 1 is saying lignin forms an incomplete secondary wall. This is a complete lie. And number 2, there is no cross walls between the vessel elements. This is correct. We do not have cross walls between vessel elements in the xylem vessels. And again, that helps to reduce resistance of water flow. Right. Then on the third response, we have the xylem vessels form narrow tubes. This might be true, but I don't see it helping to reduce the resistance of water flow, and therefore I would go with 2 only, which is C. Now on number 28, it says, what causes water to move from the root cells to the endodermis? What causes water to move from the root cell to the endodermis? I hadn't gotten the question the first time. So now going through our answers, A is saying diffusion through cell walls and osmosis down a water potential gradient in the cytoplasm. This looks just fine for the movement of water from root cells to the endodermis. It looks just perfect. But let's go through the other options. Diffusion through the symplast and osmosis and root pressure through the apoplast. This answer is not so accurate, because diffusion through the simplest way only deals with the movement of solids and not water. So we cannot safely say diffusion through the simplest when you're talking about water, and therefore B is wrong. And that's one part of it that's wrong, but I didn't even bother to look at the other parts. Then C, osmosis from cell vacuole to cell vacuole. So C is suggesting that if we have a cell and a vacuole and a cell right next to each other, we have water going from one vacuole to the other by osmosis. And as we all know, that is a lie, and water does not move by active transport. So C is wrong. Then D, osmosis through the intercellular spaces and diffusion in the cell walls. So osmosis needs a partially permeable membrane. It cannot just happen through intercellular spaces. So D becomes wrong. And I guessed right. 28 is A. Now number 29 says, which changes occur as carbohydrate is moved into a sink? If we look at our answers of water potential of the sieve tube element, so let's say this is our phloem, right? And this is our sink. Let's say this is our source and this is our sink, right? And we have carbohydrates that are being transported in the form of sucrose. So they moved into the sink. The carbohydrates are moved into the sink. Let's assume that there was some sort of stuff or fluid in the sink. So when the carbohydrates are going to be added to the sink, if we look at this part, which says the volume of liquid in the sieve tube element, the volume decreases because we are having the carbohydrates leaving. The volume decreases. So A and C, we are having an answer from either A or C. The volume decreases because the carbohydrates are moving out. Now the water potential, because we have something like solids leaving the sieve tube element, the water potential is going to be high. Remember, the more the solids, the lower the water potential. So now we are having some of the carbohydrates moving out. So that means less solids, and less solids means a higher water potential. So then C is our answer for 29. And for number 30, says which statement correctly compares blood plasma and tissue fluid in a healthy person? Part A is saying blood plasma contains more protein than tissue fluid. This is a fact. Then B, both blood plasma and tissue fluid contain red blood cells. We know that red blood cells do not seep through the capillary walls to get to the tissue fluid, so we cannot have red blood cells in both the blood plasma and the tissue fluid in a healthy person. Of course, it can happen if someone is sick. Then C is saying tissue fluid contains white blood cells. Again, white blood cells do not filter out via the capillary walls. Then D, tissue fluid is formed from blood plasma and is not returned to blood plasma. This is not accurate because we are saying we have tiny pores in the capillary walls, which allows exchange of substances between the blood plasma and the tissue fluid. So then we can't say tissue fluid is formed from blood plasma and is not returned to blood plasma. There's some sort of exchange that still happens even after we have tissue fluid and blood plasma separately. So now our answer for this, I believe, will be A. 31. The body maintains an average normal blood pH of 7.4. Which row describes the conditions that would increase the dissociation rate of hemoglobin the most during periods of intense exercise? So blood pH and blood carbon dioxide partial pressure. It's good to notice that we're dealing with sort of like factors that reduce hemoglobin. For number 31, it says the body maintains an average normal blood pH of 7.4. Which row describes the conditions that would increase the dissociation rate of hemoglobin the most during periods of intense exercise? This question, it's good to understand that we are looking for something like factors that reduce hemoglobin's affinity for oxygen, right? And if we look at what we're given, blood pH and blood carbon dioxide partial pressure, from what we've learned, we know that it is high carbon dioxide levels and low pH that both reduce hemoglobin's affinity for oxygen, leading to an increase in dissociation, right? And using 7.4 as a reference point, if we were to lower the pH from our answers, we get 7.2. So then we discard C and D. Now, if we look at these, from here, I'm just going to pick the bigger one of these two, which is B, because I want higher carbon dioxide levels and low pH. For number 32, it says the graph shows changes in blood pressure during one cardiac cycle. Then we can see the graph says what is happening at time X in terms of the aortic semilunar valve and the atrium. So we need to determine whether the aortic semilunar valve is opening or closing at point X. And you can see that ventricular pressure is increasing. And one principle that valves use is when pressure behind them is higher than the pressure in front of them, they open. So we know that ventricles are sort of like behind the valves, the aortic semilunar valves. So then if ventricular pressure increases, they will need to be opening. And now for the atrium, if we look at atrium, it's down here, the pressure is down here, and it starts to sort of like decrease. And usually for the atrium or the atria and the ventricles, when pressure is decreasing, blood is filling. So blood will be getting into the atrium or the ventricles. And when pressure is being increased, then we know that blood is leaving the atria or the ventricles. So in this case, I will go with filling. and our answer will be D. And for number 33, says how many times must a carbon dioxide molecule pass through a cell surface membrane to travel from the tissue fluid into the blood plasma? Then it says assume there are no pores between the cells the carbon dioxide molecule must pass through. Assume there are no pores between the cells the carbon dioxide molecule must pass through. And for this, I think we need a diagram. So if we have blood plasma, which is the liquid part of blood, and a cell, which is a capillary cell, you know that the walls of the capillaries are 1 cell thick. And then on this side, we have tissue fluid, and we have carbon dioxide passing through like this. We can see that it crosses cell membrane number 1 here and cell membrane number 2 there. So for where it says how many times the carbon dioxide molecule pass through a cell surface membrane to travel from tissue fluid to blood plasma, I would say it's 2. So I'll go with B. Unless of course they had said how many times must the carbon dioxide molecule pass through layers of a cell membrane. We know that it's sort of like a double layer. We know it's like this. So then it will be 1, 2, 3, 4. But when it says how many times does the carbon dioxide molecule pass through a cell surface membrane, it's 1 and 2. Number 34 is saying 2 types of epithelial cells, 1 and 2, occur in mammalian gas exchange system in a healthy human. We have 1 and 2. It says which shows the function and distribution of these types of epithelial cells. And cell type 1 is saying absorption in the bronchi, exchange surface in the alveoli, absorption in the bronchi and exchange. Okay, it's one and the same thing. Then for cell type 2, you see there's cilia there, and to say secrete mucus in the alveoli. We do not have secretion of mucus in the alveoli, at least not in the alveoli, maybe in the bronchioles and the bronchus, but there's no mucus being secreted in the alveoli. Move mucus in the alveoli. Again, we don't talk about mucus, or we don't put mucus and alveoli in the same sentence. So here it says move mucus in the bronchus. Move mucus because of these cilia hairs, and so exchange surface in the alveoli. I would take D to be our answer, because the same is 1 for B. So the only defining part was secrete or move. But we can see that we have the cilia hairs, which usually act in moving mucus. And for the 35th question, we're saying which tissues would be seen in a photomicrograph of the wall of a trachea. And we have elastic fibers and smooth muscles. So both of these can be seen in the trachea. So our answer is C. So this is like a rephrased question, which originally used to sound like which structures do you find in the trachea. They twisted the wording a bit and they gave you in form of microscopy to say, if you look through a microscope, what features do you see? And they give you examples, elastic fibers and smooth muscles, to further confuse you. But just remember that elastic fibers and smooth muscles are both found in the trachea. And now 36 says the graph shows a decrease in cases of tuberculosis, TB, in a country between 1910 and 2000. It says which factors could have contributed to the fall over this period? 1. Pasteurization of milk. This is well correct. Then 2. The provision of new housing to reduce overcrowding. Since TB is airborne, we do not need overcrowding, so this might be a good choice. Then 3. Removing large pools of stagnant water. We know that's for malaria, so we can take that for TB. And lastly, identification of contacts of people infected with TB. This again is correct, because if people are identified, then they can be isolated and the general public will be safe in course. So then we take 1, 2, and 4, and that's A. And now on 37, it says the pathogens for some diseases are transmitted by vectors. Which disease is transmitted with the help of a vector that could be controlled by removal of pools of water? We've just talked about that here in the previous question, and that is malaria, of which the vector will be the female Anopheles mosquito, which lays its eggs in stagnant water. So if we remove the pools of water, then we can reduce it. And for 38, it says how does penicillin affect bacteria? Part A is saying it inhibits DNA replication. No, 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 no. It has nothing to do with DNA replication, even when it affects the bacteria. And on B, it inhibits translation. Hell no. It does not inhibit any translation or anything to do with DNA replication, translation, transcription, and stuff like that. Then C, it is a competitive inhibitor of an enzyme in the cell wall synthesis. Now, that's correct. It deals with the cell wall, right? It affects the wall, and then you can end up having holes or loose spaces in the wall, and when water gets in via osmosis, the bacterial cell will burst, and that's how they work. And D says it is a competitive inhibitor of an enzyme in protein synthesis. Again, no protein synthesis, and also nothing to do with DNA translation, transcription, and all that. It deals with an enzyme in the cell wall synthesis. And so we go with C. Now for number 39, says the hybridoma method is used for the production of monoclonal antibodies. Then it goes on to say which two types of cell are used in this method. Not too sure about the wording there, but let's try to answer it. We know that for the hybridoma method, we use tumor cells, which we also refer to as myeloma cells. And so we go with either C or D. And because we want the lymphocyte to produce a specific antibody, we go with B, lymphocyte. And so our answer will be C. And lastly, number 40, we're saying what describes a non-specific immune response. A is saying activation of killer T lymphocytes by infected cells. This is specific. T lymphocytes are specific. And B is saying cloning of B lymphocytes to form plasma cells. That does not sound right. Cloning of B lymphocytes to form plasma cells. It is wrong. Then C, ingestion of a bacterial cell by a neutrophil. Now this is not specific to any form of bacteria or in the neutrophil. It just gets ingested because it's foreign to the body. And we can go with C for non-specific immune response. And if we talk about recognition, if we start talking about recognition, just remember that that's specific. For a cell to recognize something, there's to be specific. And so ingestion of a bacterial cell by neutrophil will be an example of a description rather of a non-specific immune response. Alright, thank you guys for joining me. Please feel free to share your comments, thoughts, and suggestions in the comments below.